There is an almost imperceptible moment in which the pleasure of scratching is transformed into satiety. That exact point at which the body says enough is not the result of chance or mere custom. For years, neuroscientists have tried to decipher which internal mechanism decides that we have already obtained sufficient relief. Before continuing, if you like this content, subscribe and give it like. Your support is essential to keep bringing interesting and relevant information. Don't miss it. Now, an investigation presented at the 70th annual meeting of the Biophysical Society, held in San Francisco in February 2026, illuminates that mystery with molecular precision. This study has revealed surprising findings on how our body regulates the sensation of itching. The finding, led by Professor Roberta Gualdani's team at the University of Louvain, identifies an unexpected protagonist: the ion channel TRPV4. This channel has been linked to the perception of pain, but its role in the itching has remained in the shadow until now. The key was not to start the itching, but to know how to stop it. The study reveals that TRPV4 does not act as a generator of the pruriginous sensation, but is part of a sophisticated negative feedback system that communicates to the brain that the stimulation of scratching has been sufficient. Understanding this gear opens a promising door to the treatment of chronic itching, a disease that affects millions of people around the world. This advance could change the way we address treatments for itching-related conditions. TRPV4 belongs to the family of TRP channels, proteins embedded in the membrane of sensory neurons that function as microscopic floodgates. When activated by changes in temperature, pressure, or mechanical stress, they allow the entry of ions and trigger electrical signals. To accurately isolate the function of TRPV4, the Gualdani team designed a genetically modified murine model. This strategy allowed overcoming limitations in previous studies and fine-tuning the magnifying glass on the role of this channel in the itching. The researchers demonstrated that TRPV4 is expressed in specialized mechanosensorial neurons and also in subpopulations associated with pain and itching pathways, suggesting a more complex dialogue than imagined. This is crucial to understanding how the nervous system works. The most revealing experiment came by inducing in mice a condition that imitated atopic dermatitis. Although animals without TRPV4 scratched less frequently, each episode was much longer, revealing an interesting paradox. This phenomenon illustrates a basic principle of neurophysiology. Many sensations depend on both exciting stimuli and brake mechanisms. Negative feedback is essential to regulate our sensory responses. The finding has far-reaching clinical implications. In diseases such as eczema or psoriasis, persistent itching is a devastating burden. However, blocking TRPV4 could make the problem worse if it is not done selectively.